Thank you very much for inviting me to talk today. So I'm going to talk about a partnering project to the Human Brain Project, which in fact ran from 2016 to 2019, the funding of that, but of course some of the papers are just coming out at the moment. And the concept of this was to map brain left-right asymmetry at multiple levels, so in terms of genetic effects on brain asymmetry, at the structural and functional levels, and also in terms of the relevance to cognitive variation and disorders. And this was a collaboration between the Max Planck Institute here in Nijmegen and the Neurofunctional Imaging Group in Bordeaux and the Basque Center on Cognition, Brain and Language. So already in the 19th century, it was observed that the injuries to the two brain hemispheres can cause different kinds of loss of function. And Paul Broca famously observed that from these kinds of injuries, it would follow that the two halves of the brain do not have the same attributes. Particularly soldiers returning from war with bullet wounds to the brain was informative in that. And this is actually the brain of a famous case that Paul Broca studied with a inferior frontal damage which caused a very severe aphasia, so severe disruption in speaking ability. And now a kind of more modern take on that is here with a functional magnetic resonance imaging study of this is contrasting covert sentence generation versus just going through a overlearned list of words. So the contrast has sentence level structure versus no sentence level structure, and then what you see is that you get prominent activation in the left hemisphere, including this region that Paul Broca first observed, which is far less prominent in the right hemisphere for this task, and also some increased activation in the left superior and middle temporal gyrus as well. And in fact, many functions and behaviors are asymmetrical to a degree, related not only to language but also spatial attention and motor control, of course, where 90% of the population is right-handed, and emotion and other aspects of cognition. So in terms of brain structure, the two hemispheres show a kind of twisting, which is illustrated in this diagram here and kind of exaggerated in this diagram just to visualize it. So here we look at the cerebral hemispheres from underneath, and what you see is that the left hemisphere lies slightly posteriorly compared to the right hemisphere. And there's a crossing of the midline as well, subtly. And in this study that we did recently, we mapped brain asymmetry at the vertex level resolution, sort of high resolution anatomical resolution in the UK Biobank individuals, which I'll talk about more in a few minutes. And the red regions are those which have a greater surface area on the left hemisphere compared to the right, and the blue areas have a greater surface area on the right hemisphere compared to the left. And this happens actually very early in development. So this is already happening in the second trimester of gestation, where anatomical asymmetries are already visible around the Sylvian fissure here. So there's a lot of literature linking changes of brain asymmetry to different psychiatric disorders. I just flashed up some recent studies here of autism. This was a Enigma Consortium study that we did with data from 54 MRI data sets showing changes of cortical thickness asymmetry in people affected with autism. This study about polygenic risk for autism and schizophrenia, I'll talk a bit more about that in a moment. Here is another study from the Donders Institute, led from the Donders Institute, about atypical brain asymmetry in autism and possible relations to language impairment in autism. And autism also shows people with autism have roughly twice the rate of left-handedness compared to the general population, again suggesting some kind of alteration of the left-right axis of the brain in that condition. So for the multilateral project, there were two kind of main themes. One was in terms of just essentially mapping brain asymmetrical organization in terms of structure, function, and also plasticity, and its behavioral correlates. And then theme 2 was the part that I led, which was the genetic architecture of the brain asymmetries. And then the idea was that from the mapping work that there would be pipelines for automated measurement of brain asymmetry in large data sets, which is what we need to do good well-powered genetic studies. And then from the genetic findings, of course, we get biological insights into the developmental basis, for example, of brain asymmetry, and also we can start to look for shared genetic effects on brain asymmetries and brain disorders, which can kind of link the two in a sort of etiological sense. So these are the papers, as far as I'm aware anyway, of that acknowledge the HBP, the partnering project funding from the first theme. So this is about mapping brain asymmetrical organisation. So there's a set of studies which were led from the Basque Centre, which were studying bilingual people, or people second language learners, and how language laterality changes as people are learning a second language which is not familiar to them. And then a set of studies from the French group on really mapping in some detail the essential regions of language cortex. And I'll just put one output of that up here. So in this study, the French group were interested to ask what are the kind of core regions of left hemisphere language network which are activated in sentence level language tasks, regardless of the modality in which the task needs to be performed. So kind of separating out the lower order regions specific to the modality, and asking the question, which regions show left-way asymmetrical activation during sentence level tasks versus word lists, regardless of whether it's sentence production task or a sentence level reading task or a sentence level listening task. So supramodal language areas in a set of right-handed individuals. And this is what they observed. And then they used resting state fMRI in the same individuals to kind of define sub-networks of this overall language network, including one kind of sentence level core network, which they defined. All right, but I'm a geneticist, so I'm going to talk mostly about the research of theme 2, which was the genetics of brain asymmetry. And so the question is, how does the brain become asymmetric in development? And so an interesting point is that this is not obviously linked to the left-right axis of the visceral organs. So people, of course, the vast majority of people have the heart located on the left side of their body, and the intestines loop around one way rather than the other way that they could have done. What's interesting is that people who have a very rare genetic condition called situs inversus of the viscera, so they have a mirror reversal of the visceral organs. But they seem to have the normal rates of left-handedness and left hemisphere language dominance. So it suggests that there's a kind of fundamental developmental dissociation between the left-right patterning of the body plan and the left-right patterning of the brain, which leaves us in the position where we don't know the essential genetic molecular mechanisms which pattern the left-right asymmetry of the brain in development. And so we wanted to do genetic studies of this, obviously to get some clues into what's going on there, and also to be able to study genetic overlaps with psychiatric traits. So this is the output from theme 2. The genetic studies, and I'll take you through a couple of these in a moment, the sort of larger scale ones that we've been able to do in recent years. But we also had some work, for example, trying to link situs inversus visceral genes with handedness, and again we found no clear link, sort of confirmation in that study. All right, so we've started to use the UK Biobank data a lot. I don't know if you know of the UK Biobank, but it's a very large population data set collected in the UK. And so, for example, for this study, which I'll talk about, we were able to use data from 32,000 adults from the UK Biobank data, with both brain MRI data and genetic data. And in terms of brain measurement, what we've been doing here is using the FreeSurfer automated cortical parcellation method and subcortical volume measurement as well as part of the pipeline. And this basically delivers regional measures of a set of cortical features which can be either regional surface areas or the cortical thickness of those regions. You see that kind of illustrated here, where the area is just, as you'd expect, the area of a region, and the thickness is the thickness of the grey matter. And also the subcortical volumes. And of course the point is that these measures have left hemisphere and right hemisphere counterparts, and then from the left and the right data we then calculate an asymmetry index for each individual in the data set and each region defined in the atlas as the difference between the left and the right measurement adjusted for the average of the left and right measurement. And so in these 32,000 individuals we find what's called a SNP-based heritability for these different asymmetry measurements of up to around 10%, which means that essentially people who are more genetically similar over their entire genome tend to have slightly more similar brain asymmetries than do sort of more genetically unrelated people. In other words, that's evidence that the genome is affecting these asymmetries to an extent. So we're then able to do what's called a Genomewide association scan, or GWAS, of these brain asymmetries, and we do this in a multivariate context here, where we're essentially, so if you look at the x-axis of this plot here, this represents the genome spread out from left to right, so like the larger chromosomes on the left, so chromosome 1, chromosome 2, chromosome 3, all the way up to the chromosome X here. And each dot on the graph represents a single polymorphism in the genome, so a site which varies between individuals, and then we test each polymorphism in the genome for its simultaneous multivariate association with the heritable asymmetries of the brain. And so there are, we're testing here millions of polymorphisms across the genome, and obviously you need to apply a suitable multiple testing correction threshold, which is here, and you can see a number of regions of the genome which are standing above that quite significantly, including this very top hit here, which has an incredibly low p-value, and we had to break the axis here to show that one. So around 21 genomic loci which are associated with variation in adult brain structural asymmetry, and then this kind of hairball illustrates how, like for a particular genetic polymorphism on here, which aspects of brain asymmetry it's actually associated with by following the lines. So you can see that there are some polymorphisms which affect multiple aspects of brain asymmetry, and there are some regions whose asymmetry is affected by multiple genetic polymorphisms. So what kind of genes are implicated by this analysis? So one of the interesting things is by then looking in postmortem data, gene expression data. So here you're measuring messenger RNA levels of different genes across from samples across the lifespan, in fact. And what you see is that the genes which are associated with the variation in adult brain asymmetry tend actually to be most strongly expressed in the prenatal brain. So in early prenatal, mid-prenatal brain, and then their expression dropping off actually into infancy and through to adulthood. So what this is suggesting is that although these genes are identified as affecting the variation, inter-individual variation in adult brain asymmetry, they're most active in the developing brain, and so that suggests that these inter-individual variation that we see in brain asymmetry is somehow set down very early in life. There's a kind of functional enrichment for these genes. So around half of the 20 genomic regions that I showed you actually are pointing to what are called microtubule-related genes. And the microtubules basically form the protein skeleton of the cell, which has, of course, a wide variety of functions. It controls the cell shape, its movement during development, internal transport within the cell, growth, axonal outgrowth. All sorts of functions are related to the microtubule. So why might this be specifically interesting for brain asymmetry? Well, so this is a hypothesis. This isn't proven, what I'm showing you here, or well, some of it is proven. The question is, how do you, very early in development, so for example, for the visceral asymmetry, right, very early in development, two of the body axes form, the anterior posterior and the dorsal ventral axis. And then the question is, how do you get a consistent third axis, the left-right axis, with respect to those two, which is consistent in direction? And for the left-right axis, the key to this seems to be in the asymmetry of the amino acids themselves, right? So amino acids are chiral molecules. It means they can exist in two different mirror forms. And if you synthesize them in the laboratory, you get a mixture of the two mirror forms of amino acids because of the way the other groups are organized around the central carbon atom. But as it happens, all of life on Earth is built on only one of these two forms, because you can only string one of the two forms together to make the polypeptides and the proteins that we're made of. And I guess some kind of accident early in the origin of life on Earth favoured one, or was based on one and not the other, and that's what we're all based on from then on. And then that essentially creates an inherent asymmetry in the proteins which are built from amino acids. And in the case of the visceral asymmetries, this is related to asymmetries of cilia, which are organelles on the surface of the cell, which spin around one way rather than the other way, and they create a kind of leftward flow of fluid within the very early embryo, and that triggers asymmetrical gene expression, which creates differences of the left and right developmental fates of the organism. But as I told you, the brain seems to be somehow dissociated from the visceral asymmetry, and now we're implicating microtubules rather than cilia here specifically. And in other organisms, including the fruit fly, in fact, they also get developmental asymmetries in their intestines, which originate not from cilia, but from kind of asymmetrical twists of cellular shape. So the cells in the developing epithelium of their hindgut take on a kind of asymmetrical twist, which is due to the cytoskeleton within them, and that seems to initiate from within the organ itself an asymmetry of development. So that's interesting, and perhaps a similar mechanism goes on in brain development as well. All right. So this is just showing that. So we're able, in these 30,000 individuals from the UK Biobank, in combination with data from other genome-wide scans for psychiatric disorders, we're able to assign what's called a polygenic score to each individual in the UK Biobank, which is an indication of their genetic risk for developing a particular psychiatric trait. So these 32,000 individuals, there's a whole range of polygenic scores. Of course, some people are genetically at lower risk of developing disorders, and some are at higher risk. And then this is a kind of map of how brain asymmetry across these 32,000 people relates to their polygenic risk of developing autism. Not that they have developed autism, but that they carry a combination of genetic polymorphisms across the genome which are associated with an increased risk of that disorder, as assessed in other data sets. And likewise a loading map of the schizophrenia polygenic risk against brain asymmetries. And so both of these polygenic risks are associated with multivariate patterns of brain asymmetry, individual differences in that. But in fact, neither of these polygenic risks was associated with a more male-like or female-like pattern, which relates to a prominent hypothesis in the literature about the kind of biology of autism. In this study, we were mapping the cortical correlates of left-handedness. So within this data set, there were 28,000 or so right-handers, and there were 3,000 left-handers. And so what we're doing here is we're measuring the asymmetry of the cerebral cortex, anatomical asymmetry, and then relating that to handedness within this sample. And so what we find, so this is the kind of unthresholded map of regions which show differences in their asymmetry according to handedness. And then this is the thresholded map for significant clusters. And so what you see is some kind of insular regions here, and some anterior cingulate regions here. This is in terms of surface area asymmetry, and this is in terms of cortical thickness asymmetry. And here, again, here we're seeing a region of kind of primary somatosensory cortex, and here more of a visual area. And so what's interesting here is that all 10 of these clusters, they actually show a rightward shift of asymmetry in left-handers. So what that means is that they're relatively larger or thicker in the right hemisphere in left-handers, which is consistent with a kind of relative shift of neural resources towards the right hemisphere in left-handers who are using the right hemisphere for controlling their dominant left hand. So that kind of makes sense. And then we ask the question, can we, so what kind of functions are these regions implicated in? Right, and so one way of doing that is just kind of going to the literature in an ad hoc way and looking up studies and saying, oh yeah, this region is important for this function, or this region is important for that function. We try to do that in a more systematic way where we use what's called the Neurosynth Database, which is basically a kind of meta-analysed functional magnetic resonance imaging data from thousands of fMRI studies. So what we see is regions that show altered asymmetry of surface area in left-handedness. They tend to be particularly important for executive functions and language-related tasks. And that's interesting because there are sort of evolutionary and developmental theories of hand motor control being left hemisphere controlled and language being left hemisphere controlled, and that these two may have some sort of co-evolution and co-development. And then in terms of the thickness, regions that show differences of cortical thickness asymmetry in left-handers, well, that you see what we get here is functional annotations, which are very sort of handy, right? So there's hand, fingers, sort of tapping and these kinds of annotations, and that's because of this particular region of somatosensory cortex which is showing up here. So that's a kind of quite nice confirmation, I guess, of these results, as well as some visual annotations based on the visual region, which shows some evidence for alteration in left-handers. Then we could also ask the question, so these are the regions of the cortex whose asymmetries are different in left-handers. Which genes, which genetic polymorphisms are they influenced by? And so, to cut a long story short, what we find is that the surface asymmetries of language-related regions are significantly heritable, and they tend to be associated with handedness-associated loci. So, in other words, there are genetic polymorphisms which affect both the probability of being left-handed and the structural asymmetries of regions of the cortex which are associated with left-handedness. Again, consistent with this idea that handedness and language might share genetic, developmental, evolutionary links. And then interestingly, the thickness asymmetry of the primary somatosensory cortex was not heritable. So that suggests perhaps that the thickness asymmetry of this region here, which differs in left-handedness, might be more of a downstream consequence of hand preference that gets established as a consequence of being left-handed rather than a sort of primary driver of being left-handed. It's just very briefly to mention that the multilateral work was kind of simultaneous with a whole bunch of work that we were doing through the Enigma Consortium, where we were mapping brain asymmetries in a whole bunch of different disorders in large-scale studies. And anyone who's interested in joining Enigma Laterality Consortium studies, please get in touch. And there are other things which are still going on at the moment, and other things in planning. All right, so just to conclude then on the genetics of brain asymmetry, at least, so these large-scale studies have finally started to answer some long-standing questions about human brain asymmetry, including which genes are associated with brain asymmetry, which particular brain regional asymmetries are associated with handedness or psychiatric traits, and how strong are those associations, and started to identify kind of three-way associations between genes, brain structure, and behavioural variability in terms of handedness. The genes that affect adult brain asymmetry are especially active during embryonic brain development. Not only, but kind of especially, suggesting that much of the individual variability in adult brain asymmetry is set out very early in life. And then we have a particular sort of functional enrichment among the genes for microtubule involvement, which hints at a potential mechanism of establishing the left-right axis of the brain. So just some practical issues. Since the project was conceived back in 2014, and I think the grant submitted in 2015, in the time in between, of course, the UK Biobank kind of came along and sort of changed a lot in the way that the samples that we would decide to focus on for the project. So the UK Biobank offers a very large sample for genetic discovery, but it doesn't have language task fMRI, unfortunately. But it meant that we redirected some of our efforts to the genetics of brain structural asymmetry because of this availability of this very large data set for well-powered genetic studies. And we're currently working on genetics of resting state connectivity asymmetry in this data, and also rare genetic mutations, which is something that's come through from exome sequencing analysis of these same individuals. Something that we wanted to do in the grant but couldn't really do at the time was because so large-scale genetic studies of reading and language performance have only just become published this year. And there are two studies here which I'm highlighting. One large genome-wide screen for dyslexia, and another for reading and language performance in thousands of people. And these data now allow us to look for genetic overlaps between brain asymmetries and reading and language performance in a way that we kind of would have liked to do earlier, but has been sort of limited until now. And then we're setting up a new research line with the mouse as a potential model for looking at brain asymmetry and its genetic contributions, looking at knockout lines for genes implicated by the human work with various transcriptomic and other techniques. So in terms of the e-brains, we haven't used it so far for the project. So for example, the UK Biobank data can't be deposited there. That's just a limit of the data, but perhaps suitable for future mouse work. And that's it. Thank you.
Thank you very much for your